First, I wanted to talk about characteristics of contractile filaments within our muscle cells. So muscle cells are sarcomeres, and right here I've drawn out a very simplified illustration of a muscle cell. Imagine if we took this and just multiplied it and just went down. That's what we're looking at here, but I just cut it into a portion. So we have these Z discs that go inwards, and on the Z discs we have nebulin and titin. These are just a couple of the things that are involved in the structure of our muscle cells. I really want to focus on nebulin because nebulin has actin that is attached to it. And actin is where we're going to be getting our myosin, which I'll talk about here in a second. But I've zoomed up a little bit on just one of the nebulin. The orange spheres are actin, and these blue lines are going to be our tropomyosin, which is a protein that is currently covering active binding sites for our myosin to attach to the actin. So a couple of components, we have our actin, which is composed of F and G actin. We have our tropomyosin, which is that protein covering the active site. We have troponin. Troponin has three structures involved in its overall being that we call troponin T, troponin I, and troponin C. These three get its name because troponin T attaches to our tropomyosin, and our troponin C is where calcium will bind. Troponin C has four binding sites, or four affinity sites for calcium. Two of them will be low affinity, which don't always have calcium on it. And then our high affinity, hence the name, it will always have calcium because it's drawn to it. And then we have our myosin. So two primary contractile filaments that we have in our muscle cells. We're going to have our thick and our thin filaments. Our thick filaments is myosin. Myosin is composed of a head that has an ATPase site and an actin binding site, and which is what we see here, which is the I in our troponin I. It's what attaches to actin. The anatomy of a myosin has two heavy chains and four light chains that are interwoven. The heavy chains is where we are going to have our ATPase site and our actin binding site, which is going to be attached to our actin. And we also have thin filaments, and our thin filaments are going to be our actin, our tropomyosin, which is that protein on the binding site, and then our troponin. So a quick example of what's going to happen to allow myosin, that thick filament, to attach to actin. So right here we see, we'll start right here. We have our troponin T, which is attached to tropomyosin. The tropomyosin is currently covering these active sites for myosin. But what happens is calcium from our intracellular fluid is going to bind onto these low affinity sites on troponin C, which is going to cause a conformational change in our actin and in the tropomyosin that is going to expose our actin binding sites for the myosin. And when that happens, myosin is going to come in and it's going to attach onto our actin. That's a quick rundown. We'll get more into that. But those are the main important things to know about the contractile filaments of our sarcomeres. Okay, so in skeletal muscle, this is how we're going to get a contraction within skeletal muscle. So in the past, we've talked about action potentials and pre-postsynaptic neurons and all that. So what we're going to have is an action potential that's propagated down an axon. A lot of this should be review. Acetylcholine is in a vesicle. When the action potential reaches the presynaptic terminal, it's going to open voltage-gated calcium channels. Those channels are going to allow calcium to flow into the terminal, which is then going to allow snare proteins to bind and pull these vesicles full of acetylcholine down to the synaptic cleft. And this calcium is going to drive it to the surface, and it's going to open up because these snare proteins bind it down. Acetylcholine is going to exit out into the synaptic cleft. We're going to have an N1, a nicotinic 1 receptor on our sarcolemma. Sarcolemma is our muscle cell, but it's the cell membrane of our muscle cell. So on the surface, on our cell membrane, we have this N1 receptor. The acetylcholine is going to bind to that. N1 is a ligand-gated receptor. Acetylcholine will bind, opening up, and it will allow potassium and sodium to drive according to their concentration gradients. But because sodium has such a high driving force, it's really going to take up the majority of this pump. So sodium is going to enter into the cell, into the muscle cell. When that happens, these right here are more voltage-gated sodium channels. So we'll say all of these right here. And what's going to happen, we also have potassium leak channels where potassium is going to be coming out. So we're going to have a varying amount of potassium leak channels, voltage-gated sodium channels, voltage-gated potassium channels, and this action potential is going to slowly make its way into our T-tubule. The T-tubule is basically just an invagination that dips down into the sarcolemma. And within this T-tubule, we have something called DHP, which is a DHPR receptor, DHP receptor, and we're gonna have ryanodine, which sometimes we call RYR. And so this right here, this is inside the cell. This is our SR, or our sarcoendoplasmic reticulum. This is going to contain a lot of calcium. We know that calcium isn't in the ICF, like, at all. There's a ton of calcium outside, not so much inside. But within the SR, we actually have really high amounts of calcium inside what we call a calsequestrin. This is basically a bank full of calcium ready to use when this action potential goes down, triggers the DHPR gate, which then opens with the RYR, or ryanodine. And calsequestrin, calcium can leave as soon as it's prompted to. And so when it senses these gates open, the driving force is really going to pull and push calcium into the cell, which is what we see here. So our calsequestrin has our calcium. The action potential propagates down the T-tubules, open the DHP gate, or DHP receptor, which then opens the RYR, allows calcium to exit the SR into the ICF, and calcium is then going to come over, and this right over here, this is what I just talked about before. So here we have our actin, we have our troponin, we have our tropomyosin on the nebulin. And what's happening here is this calcium is coming up to our troponin. And our troponin has troponin C sites. So we had those 4 affinity sites, 2 low and 2 high affinity. This calcium that's released is going to come over to our troponin C. It's going to bind onto our low affinity sites, which then prompts the conformational shift within the tropomyosin, exposing the active sites on our actin. This right here is myosin. And right now, it is in what we call a cocked position, where it's ready to go. And when calcium exposes these sites, our ADP head that's on our myosin is going to come up and it's going to it's going to attach onto our actin. When that happens, we see here that the conformational shift happened. It's sitting here. Right here, it comes up and it attaches onto our actin. The next step that happens is when it attaches, this ADP and phosphate are going to come off. And then what's keeping this myosin cocked and ready is this ADP and phosphate. So when this comes off, this is what we're going to call a power stroke. Now it's in this uncocked position because it's binded onto the actin. And it's going to stay there until it has enough energy to pull itself away from the actin. And what's going to help it do that is ATP coming in and binding onto the head, which gives it enough power to push itself away from the actin. And when that happens, it detaches, it hydrolyzes back into ADP and a phosphate. And after that, this is our recovery stroke. So it goes from uncocked and this back to its cocked position, ready to go. That is what happens in our skeletal muscle when we need to contract muscle. And it all starts with this action potential, which then opens up calcium voltage gates, which lets acetylcholine out, which then opens our N1 receptor, which then allows sodium into our the muscle cell, propagating the action potential down the sarcolemma into the T-tubule, hitting the DHP receptor, opening RYR, letting calcium from the calsequestrin out of the SR into the ICF, coming over, attaching onto our troponin C site, which causes a conformational shift, which allows myosin to attach, which allows contraction to happen. So when stimulation stops, this will keep going on as long as we have calcium. So when this stops and our myosin is hydrolyzed back into its cocked position, calcium is going to be actively transported back into calsequestrin. It's filling the bank right back up. And it does so by what this little receptor right here. I kind of drew it small, but basically this, I said receptor, this gate is going to act, it's a primary active transport, and it's going to take calcium, because calcium wants to be out here and drive down, that's its concentration gradient. This pump is going to push it against its gradient back into calsequestrin. This pump right here is highly dependent upon ATP, and it's actually the highest consumer of ATP within our muscle cells. And this pump is called our circa pump. All we need to know is that it uses ATP to push calcium back into the calsequestrin against its concentration gradient. And when that happens, calcium is then no longer attaching to our troponin C, which means that tropomyosin is going to block the active sites again, which means our myosin is no longer able to connect back to the actin, which means it's in its relaxation state, ready to go again, prepped and ready. Okay, so now we're going to look at 3 sources of energy that we use for contraction, because if you recall our cycle, the cycle that we just did is called the cross-bridge cycle. So the cross-bridge cycle obviously needs ATP to continue. And so these are sources of energy that our body uses to receive that ATP. So there's 3 ways. I'm going to talk about 2 first, because these 2 don't need oxygen. And the last one is aerobic, so it's going to require oxygen. So we'll just talk about these 2 first. So our first one we're going to talk about, this is creatine phosphate. If you go to the gym, if you're used to that, a lot of gym goers will take creatine for various health reasons that help. And this is going to explain, hopefully, why taking that as a supplement is pretty beneficial. But creatine phosphate, the energy it provides is pretty limited. So if you are picking something up or you're doing a very short burst activity, you're going to be using up energy created by creatine phosphate. It's limited, so it lasts 10 to 15 seconds, or it helps contract for 10 to 15 seconds. The mechanism by which it happens, it donates a phosphate to ADP, which generates ATP. So obviously, the more ATP you make, the more energy you're going to have. That's basically how it does. It just donates a phosphate. An advantage to this source of muscle contraction and skeletal muscle is its immediate ATP production. So it's when you need it, on the spot. And again, it doesn't require oxygen, so that's a plus. A disadvantage is it depletes very fast. Again, it's limited 10 to 15 seconds. So that's a big disadvantage, doesn't last. Again, it's used short bursts of energy. That's when we're going to use it. Next, we're going to talk about anaerobic glycolysis. Glycolysis refers to glucose. This source of energy is going to be able to sustain contractions for about 30 to 60 seconds. So it's a bit longer than our creatine phosphate. The mechanism, it breaks down glucose into pyruvate. Pyruvate, every glucose we get 2 ATP from it. A downside to breaking down glucose into pyruvate is that we also produce lactic acid, and that lactic acid will lead to muscle fatigue. So it comes with a negative, so keep that in mind. But it is nice because obviously we eat glucose, and being able to break that down into energy to contract our skeletal muscles is really good. Advantage, it's also pretty fast. It's really rapid ATP production. So that's a plus. And again, it's anaerobic, so no oxygen is required for that. A big disadvantage, again, is the muscle fatigue that comes with the lactic acid produced by the pyruvate. We use this for moderate bursts of activity. So sprinting, like a 100-meter dash, things like that, we're going to be using this quite a lot. So next I'm going to talk about one that requires oxygen. So yeah. Okay, so this last one is going to be aerobic or oxidative metabolism. Its availability for very prolonged activities. So this lasts for hours, whereas our first two was either 10 to 15 seconds or up to a minute. This one goes for hours, as long as there's fuel available, glucose. So the mechanism is glucose and fatty acids, amino acids, they're oxidized via the Krebs cycle. This produces 36 to 38 ATP per glucose. So definitely this is the most efficient source of energy. It's a good thing because there's also no production of lactic acid, so fatigue, not as much with this. Disadvantages, it's slow, takes time, it requires oxygen and mitochondria. That is something the other two did not require. It's very important. And we use this for long durations like marathons, hiking, all that. So there's your 3 sources of contractions. 2 main types of smooth muscle. So everything we talked up to this point mainly relates to skeletal muscle. Right now, we are going to dive into smooth muscle. So there's 2 types. We have single unit and multi-unit. A couple of attributes I've written out. Single units are coordinated contractions, and they're rhythmic. We see this in our intestines as it pushes stuff down our intestines. Each house strut contracts one at a time. It's rhythmic. We see this in hollow organs, so like I said, intestines. We see this in bladders, uterus, things like that. Our second type is multi-unit. This is more precise control, and it's individually innervated, which is different from single unit. We see this in large arteries, our bronchioles, and things like our iris and our eyes. This requires direct or hormonal, so direct neural or hormonal stimulation, and it's not rhythmic, unlike the single unit. So those are 2 types. Now we're going to dive further into smooth muscle. Okay, so now we're going to look at how smooth muscle contracts. I'm going to pull it up on the iPad because it's pretty tedious, and it's better if we can look at this chart that I already have drawn up. So there's going to be 2 types of smooth muscle contraction. We have phasic, which is rhythmic contractions, and we have tonic, which is going to be sustained contractions. So here you can see we have our intracellular calcium that we just talked about right here. That's going to be coming from that IP3 opening our ryanodine. So that calcium is going to come into the intracellular fluid. We have this right here, our calmodulin. Calmodulin has 4 binding sites for calcium. So for calcium, going to bind to our calmodulin. It's going to activate that. Here we have MLCK. What calmodulin is going to do, right now you see that it has this bit stuck. It looks like a snake and its tail is in his mouth. This calmodulin is going to activate
our MLCK. MLCK has a phosphate. And when our myosin attaches to our actin, like what we talked about earlier with skeletal muscle, when that myosin comes in, this phosphate from our MLCK is going to attach onto our myosin. So now our myosin not only has its normal phosphate and the ADP, but it also has this extra phosphate which helps it attach to our actin. The next step, what follows, a phosphate and our ADP are going to leave right here, step 4. And this is where our power stroke is initiated. And if you notice, unlike in the skeletal muscle, we still have a phosphate attached during our power stroke. So this is going to return that myosin to a lower energy state. And it's going to be in its uncocked position right here. And the next step is we're going to have ATP come back into at step 5. And right up here, you can see that ATP attaches, and just like how it pushes it off of actin and skeletal muscle, when it attaches, it's going to do the same thing, and it's going to dissociate from the actin. This allows our myosin to cock again, back into its cocked position. And after that, we have another thing over here that instead of MLCK, this is going to be MLCP. And MLCP is interesting. So rho kinase phosphorylates, meaning when rho kinase is active, our MLCP is going to be active because there's no phosphate. When we have a phosphate that's blocking this site right here, it's going to be an inactive MLCP. So if there's no phosphate, it's active. If there's phosphate, inactive. Right here, we have an active MLCP, and that MLCP is going to come in and it's going to knock this phosphate off of the myosin. So now by the time we come back up here during our relaxation phase, our myosin is going to hydrolyze again. So this ATP right here is going to turn back into ADP and its phosphate right here. And now we're back at the beginning, ready to go again. And that is how we go through our phasic smooth muscle contractions. Okay, so next we're going to be talking about tonic. Something important to remember, this MLCP right here, highlighted in red, we see that if rho kinase is available and active, starting right here, if rho kinase is available and active, it will put a phosphate on the MLCP to inactivate it. However, if rho kinase is not available or it is inactivated, then MLCP will be active to do its job of removing the MLCK phosphate. Now, what activates rho kinase? Calcium. Calcium is what activates rho kinase. So when we have a buildup of calcium, that means that there's going to be more rho kinase to inhibit our inactivate our MLCP. That's what we're going to see with tonic. You can see here we start the same. We have our myosin head, ADP, and a phosphate. Calmodulin comes in. Calmodulin attaches to calcium. That calcium then activates our MLCK. MLCK binds. We have another phosphate here on our myosin. Now, extra MLCP is going to remove this phosphate. So as before, right here, we're just removing this phosphate and an ADP. Here, we're going to be removing everything. This is because we have an excess buildup of MLCP that's going to come in. And instead of removing the phosphate here in step 7, it's going to do that early. And when that happens, our myosin head is going to attach, and our ATP is going to come in, and it will attach again to its myosin. But this right here, our latch state, is a slowing rate of dissociation of the myosin and the actin. Our absence of MLCK interferes with myosin's ability to dissociate from actin. It happens, it's just a lot slower than it would be here in our phasic rhythmic contractions in our smooth muscle.
Okay, so now I want to compare, up to this point, everything we've talked about between skeletal and smooth muscle. So I have some categories here. First, we'll compare about structure. We'll compare the control, what controls it, the regulation of contraction. We'll talk about where it gets its calcium, how energy efficient is it, its tension regulation, and then how prone to fatigue is it. So, starting off, skeletal and smooth structure. Skeletal is long and it's striated, but with smooth muscle, it's more of a spindle shaped, which we'll get into here in a second, and it's not striated. What controls skeletal, it's all voluntary. So our somatic nervous system, anytime you move, that is skeletal muscle, things you can voluntarily control. Smooth muscle is involuntarily. So a lot of our autonomic nervous system and hormones, things like that. Next, regulation of contraction. So you saw the cross bridge cycle that I drew for skeletal muscle, and then we just got done looking at it on my iPad. Biggest differences. Skeletal calcium comes in binding to that troponin C, which then attaches to the active site on actin, and moving tropomyosin out of the way, causing that conformational shift of tropomyosin. For smooth muscle, the calcium comes in and is going to bind to our calmodulin, those four sites, and it's going to activate our MLCK. That's a big difference on how it starts. And the source of calcium for these. So for our skeletal muscle, probably like the majority of the source of calcium that we're going to get is all from that SR, that sarcoplasmic reticulum. For our smooth muscle, it's going to be a lot from the ECF, calcium from the ECF, while also from the SR. If you remember that IP3 from our G protein coming in, activating our ryanodine, allowing that from the SR to go in. But we also have all these other factors from the outside because we have a ton of calcium on the outside and a lot of means to push it in. So smooth muscle is really going to do what's most efficient. If it's more efficient in that circumstance for IP3 to bind to ryanodine, allowing SR to push calcium in, it'll do that. Other means, by getting calcium in, it's going to do that as well. This is really focusing on efficiency. Speaking of efficiency, skeletal is fast and our smooth is very slow. That makes sense when we think about, if we need to grab something, reflexes, which we'll get into here in a second. All that, skeletal muscle has involuntary aspects to it, but we can control them. So that's a big difference too. Skeletal has a really high ATP usage, whereas again, smooth muscle is very energy efficient. We saw this with that latch method where the myosin stays in its uncaught position for a while, and it slows down that cycle until more calcium comes in to activate the rho kinase to inactivate MLCP, which allows the cycle to continue. That's beneficial. It's not a bad thing, and it's very energy efficient. That's a big reason why we see it. Fatigue, skeletal muscle, it's all or nothing. It gets really fatigued. Whereas smooth muscle, it can contract for long periods of time. Again, in our tonic phase, which we saw with the latch, we're going to see very long periods of contraction, and that makes complete sense when we think about, our intestines and everything that smooth muscle does. So those are the main differences. Now we're going to talk about reflexes a little bit, and then we're going to finish up.
Okay, we're going to finish up here on my iPad just so we can get a more in-depth look at, we're going to look at this muscle spindle. After that, we'll look at a Golgi tendon, organ, and we'll look at stretch reflexes and a withdrawal reflex just to finish up this essay. So let's start by looking at this muscle spindle right here. Muscle spindles are embedded within the muscle belly, and it's parallel to skeletal muscle fibers. It's composed of intrafusal fibers and extrafusal fibers. So spindles are going to be proprioceptors that monitor muscle length. They are basically bundles of skeletal muscle fibers with sensor and motor innervations. The extrafusal fibers on the outside, they're going to make up the bulk of skeletal muscle, while our intrafusal fibers are going to run parallel to them. Note that extrafusal fibers are, I can't say that word, are contractile. So muscle spindle's main functions is going to be to sense muscle stretch and to signal that to the central nervous system. It's also going to activate stretch reflexes, causing contraction of the same muscle to resist it overstretching. And this is modulated by gamma motor neurons, which help adjust the sensitivity. When muscles aren't stretching, what could happen is that they could droop quite like this rubber band right here, or they could sag. To prevent the sagging of these muscles when they're not being stretched, that is when our gamma motor neurons, which innervate each spindle at the contractile end. And so what's going to happen is, to prevent the sagging, you can see here that we have all this excess. These gamma motor neurons are basically going to move their way just enough inwards to where we could put constant tension on our muscles right here. This is going to make it so that when we need to stretch our muscles, if they were sagging constantly, that would completely get rid of the use of our muscles. So our gamma motor neurons that keep some tension on them make it so that they're always ready for action. And that is the most important part of the gamma motor neurons prevent these sagging. Our spindles that we've talked about, spindles are going to only respond to stretch. That's why it's important to have our gamma motor neurons right here that are going to keep tension, so that we can respond to stretch when it happens, because if they were sagging and a force starts to push outwards either of those ways, it's going to take a long time to detect any stretch at all because it's sagging. So the gamma motor neuron is going to help our spindles respond to stretch. The next thing we're going to talk about is our Golgi tendon organs, which respond to tension. So spindles respond to stretch, and our Golgi tendon organs will respond to tension. So just an example of our spindles that sense stretch and the importance of our gamma motor neurons detecting that, or helping it keep ready to detect a stretch. Here, a good example of when you go to the doctor and they hit this tendon right here, and your leg's going to shoot up. This is because when the doctor hits this tendon right here, our spindles, these are going to detect stretching. And when that detects stretching, sensory neurons are going to create an action potential in our quadricep muscle right here. This is going to go up all the way to our spinal cord. It's going to enter into our spinal cord. And sensory neurons are going to synapse with the alpha motor neurons and inner motor neurons. So every reflex, we're going to inhibit a muscle as we contract another one. So we can see here, after this action potential comes into our spinal column, we're going to get a EPSP and an IPSP. These are going to be excitatory and inhibitory. The excitatory signal is going to come out of the spinal column back and over to our quadricep muscle, contracting that muscle, while at the same time sending an IPSP all the way to the hamstrings to inhibit it. We can't have two muscles that work against each other to contract at the same time if we're trying to react to these muscle spindles sensing stretch. So when we detect the stretch, sends a signal to our spinal column. In there, we have our alpha motor neurons, or some alpha motor neurons, while we also have inner neurons. Inner neurons are going to be what turns this right here into an IPSP and an EPSP. So that's going to come, it's going to contract our quadriceps to move with the motion to prevent overstretching, while this IPSP right here is going to come and inhibit our hamstring. Now, for the Golgi tendon reflex, we just got done looking at the stretch reflex. Again, spindles respond to stretch, while this Golgi tendon reflex is going to respond to tension. It's going to be pretty much the opposite of what our stretch reflex is. Stretch reflex, it's excitatory, and it contracts the stretched muscle group. But our Golgi tendon reflex is inhibitory, and it's actually going to relax the affected muscle. The main purpose of the Golgi tendon reflex is to prevent injury, and it's going to help regulate our muscle contraction force, because our muscles are actually strong enough to put so much pressure on our bones that it can break our bones. And so it's really important to have this limit that our body's made to prevent that from happening. So our Golgi tendon reflex here is going to sense tension. So the stretch reflex sensed stretch, but now there's going to be tension here, and that tension is going to send an action potential all the way back up to our spinal column again. It's going to synapse with inner neurons and with alpha motor neurons. And you can see here that wherever it sensed tension, it's going to have a IPSP and an EPSP. Again, and it's going to go to our quadriceps and have it inhibited, while the alpha motor neuron is going to go to the hamstring and it's going to contract the hamstring while the quadriceps relax. It's very similar to stretch, but again, the main difference is that it's responding to tension, and this is to help prevent injury. So wherever it detects this tension, it's going to send an inhibitory signal to that quadricep, and it's going to contract the other muscle to prevent this from being injured. So lastly, we're going to look at a withdrawal reflex. A good example of this is stepping on a nail or stepping on a Lego or slipping on ice. Basically, the same thing's going to happen. The idea here is that when we have a stimulus, it's going to go up to our spinal column again, and the body, both sides of the body are going to work with each other here. So this right side of this person's foot is going to want to come up. And as that comes up, our left side of the body knows that it has to compensate for the shifted weight. So as these flexors on his right leg contract to move his foot up, our left side is going to relax those flexors. And whatever flexes on this side is going to relax on this side, and vice versa. Whatever contracts on this side will relax on this side, and this all happens right here. Stimulus and inhibition signals. This is called reciprocal inhibition. So when one side of the body reacts one way, our other side is going to be inverse to that to help compensate for whatever stimulus it is that drove us there in the first place.
Quick recap to finish off. We're talking about stretch reflexes and Golgi tendon reflexes. We have our muscle spindles for our stretch reflexes. Can't see that spindle. And then Golgi tendon organs. I just wrote down the differences between the two really quick to finish off. Muscle spindles are in the muscle belly, whereas our Golgi tendon organs are going to be at the site close to where the tendon and the muscle attach. This is going to activate our stretch reflex, which is contraction, and our Golgi tendon organs are going to activate an inverse stretch or muscle relaxation. This is to prevent from damage and excessive force, whereas our muscle spindles are going to resist overstretching. Again, stretch reflex is in response to stretching, and Golgi tendon organs is the opposite of that, and that's to prevent the damage from those muscles and parts of our body. So there's no direct innervation for our Golgi tendon organs. It's more like a receptor itself, and our muscle spindles, again, modulated by gamma motor neurons. That is to prevent our muscles from sagging, to keep them constantly ready, primed, ready to go, to detect stretch. That is the whole essay. Everything I need to talk about. Yeah.